Well, thanks very much, Henry. I really appreciate it. Thank you not only for organizing today's forum, but for organizing this whole series. It's really a great idea, and I look forward to not only my participation, but I've already enjoyed hearing my other colleagues. For any of you who are participating from outside the Stevens community, this is a photograph taken from an airplane going into Newark Airport, and this is downtown Manhattan, the Hudson River in the foreground, and the Stevens campus is right on the western bank of the Hudson River. So it's really an exciting place to be. My research has to do with biomaterials associated infection, and this is a problem that is significant, to say the least. And it comes because, as you can read here, tissue contacting biomedical devices provide surfaces where biofilms, bacterial biofilms, can grow, and it produces chronic infection. And usually this infection can't be resolved in any other way except by removing the device. And it can have significant consequences on both the patient and the healthcare system. You see here in the bottom left sorry, now I can't advance, there we go. You see at the bottom left an image from the cover of an old science magazine, really meant to highlight how prolific the use of tissue contacting biomedical devices is. I'll assure you that this is not a real human being. This is simply cobbled together, but there are orthopedic implants, there are shunts, there are stents that go throughout the body. You can see in the center photograph an image of a patient with an infected knee. Again, devastating, and something that's really a little known fact that I think helps put the significance of this problem in perspective. You see from the graph on the right that the 5-year mortality rate for infection associated with a prosthetic joint, a hip or a knee, exceeds the 5-year mortality rate associated with breast cancer. When I learned that, it was startling, and we'll hear more about breast cancer from one of my colleagues later in today's forum. There's a lot of detail in this slide. I won't go into it. It simply lists many different types of devices and statistics about their infection rates. The key things are really in the box in purple, that all devices, big, small, seemingly insignificant, are susceptible to infection. The infection rates, thankfully, can be relatively low, a few percent. But what's really significant is in those cases where infection does occur, it again can have significant consequences on the patient. So, you know, an obvious response is, well, why don't we just incorporate antibiotics into these systems and kill bacteria as they approach the systems? So here's one example of that. This is what's called antibiotic-loaded bone cement. Long story short, through the 80s and 90s, clinicians were simply mixing in the surgical theater antimicrobials with the bone cement and using that. You can see the cartoon in the top left to literally fix what's called a cemented hip in place. In the bottom left, those are beads that are used to fight infection. Processes were approved just after 2000 by the FDA, really as a means to standardize the process of the concentrations of the antimicrobial. The schematic graph on the right is pretty important in the sense that these antimicrobials are being released by a process called elution. They simply ooze out of the bone cement over time. And the plot that you see with the dashed line decreases over a long period of time. Early, there's a high concentration that's released, and as time goes on, there's a low concentration below the so-called MIC, the minimum inhibitory concentration, where, frankly, the antimicrobial isn't effective. There's a lot of push against these kinds of systems, though, and very few antimicrobial loaded systems have been developed in recent years, in great measure because the FDA is pushing against that. And in great measure because the antimicrobial is being delivered whether you need it or not. And if you don't need it, if you're not threatened with an infection, then having that antimicrobial present simply promotes the development of antimicrobial resistant bacteria. So the category of research that I've been working on falls into developing materials that are called self-defensive. And so the cartoon on the left shows a coating, the blue film on some sort of implant. It could be a hip, a knee, any of these things. The green color is supposed to represent a live bacteria in the so-called planktonic state. And these are much less of a threat because antibiotics can kill those. What we want to prevent, though, is those bacteria colonizing the surface. And you see that one touching the surface that's turned red, and that's because it's giving off some trigger that locally causes the release of antimicrobial. And so you can now see the cartoon on the right side that shows that traditional elution and compares it with this self-defensive approach where there's a quick burst release to kill bacteria that provide these triggers, but everywhere else on the surface, the antimicrobial remains sequestered. There are several triggers. One is pH, another is secretion of enzymes. A third one is what we're calling contact transfer, and that's the one I've been working on. And just to illustrate by cartoon the process, here's an image of a hip. It's got a very rough surface to promote osteoblast ingrowth, bone ingrowth. We've been using these things called microgels, anionic microgels. They're negatively charged. We can electrostatically deposit them onto the surface of the implant. So that's the middle cartoon. And we can do that simply by applying a positive charge to the implant itself. And that's relatively easy. One of the exciting things, though, is there's still a lot of negative charge in these gels once they've absorbed on the surface. And so we can expose them to cationic antimicrobials, small positively charged molecules, which then complex with the gels, and the gels deswell. But now we've got a reservoir of antimicrobial that can be whose release can be triggered. Not to go into too many details of the experiments we do, but one category is to follow this microgel size as it's being loaded. So this is an example of polyacrylic acid microgels deposited on a glass surface, and we're following them by video microscopy. So here on the right, you see very little contrast. There are a few microgels, but you see almost nothing because these are mostly hydrated. They're about 98% water. And when we expose them to a particular antimicrobial colistin, which is cationic, you can see they deswell very quickly. And we can follow that deswelling kinetics, and that tells us an awful lot about the state of the antimicrobial in the gel. One of the cool things about the self-defensive aspect is that in the absence of pH, in the absence of enzyme production, both of which are associated with metabolic processes, we've been able to show that just the contact between bacteria and these loaded microgels is enough to trigger the release, the local release of the antimicrobial and kill the bacteria. So this is one of the experiments we've done. Again, we're following it by in situ optical microscopy. We have a nanoprobe to which we have affixed an optical fiber. I can give a shout out to my colleague, Professor Henry Du, because he does a lot of work with optical fibers. And this is a fiber that we stole from his laboratory and cleaved. And on that surface, we've deposited microgels and loaded them with colistin. The significant thing about this nanomanipulator, we can bring the microgels in contact with bacteria on the surface. And that the result of that is illustrated here. So these green lines you see are E. coli. The fact that they're green reflects the fact that they're alive. This is before contact. When we contact the surface, so we're looking from below, the circle that you see is the end of this 125 micron diameter optical fiber. These yellow dots are the colistin loaded microgels. And you can already see that the bacteria are turning red, which is an indicator that they're dying. And after 30 minutes, all of them are killed, but only the ones that are in contact with the microgels themselves. And one important comment I want to make is in a real experiment or a clinical situation, there's no way we flood a surface with this many bacteria. So our ability to kill just those few bacteria that contact the surface is really important. Let me just conclude with a couple broader impact kind of comments. A number of us have been involved in what's called the Stevens Conference series on bacterial-material interactions. We had our 5th such conference last June, and you can see the overview photograph. One of the things that I was really excited about, we used that as an opportunity to film a short video about implant infection. If anybody's interested in this, I would encourage you to go look for it online. If you don't want to write this down quickly, when I need to find it, I simply Google on Stevens implant infection and it comes up. And I'm excited that since last September, it's already had over 1,000 views. I wish my research manuscripts got cited that quickly. Then the final thing is a project I'm working on, which is much bigger. And I'm calling it a Bhag. This is a phrase I learned about 15, 20 years ago from a colleague who used to be the chief scientist at the Army or the Air Force Research Laboratory. And preparing for this, I wondered where that title came from. Bhag stands for a big, hairy, audacious goal. So I Googled it and I found this book on the right, Built to Last, which was published in 1994. And basically is just what it says. It's a big, hairy, audacious goal. And what we're looking at is, the question of, can we eliminate this infection altogether? And it's a big project. And I'm doing this in collaboration with Da-Cheng Reng at Syracuse, Karen Sauer at Binghamton, and me at Stevens. There's a team of us doing this. We're really doing 2 parallel efforts. One is we're trying to develop a roadmap of how we might solve this problem. And 2, we're working on an NSF Engineering Research Center proposal. We've already made some pretty substantial progress. I won't go into too many details. I imagine Henry started getting nervous that my 8 minutes is probably already passed. But there are 3 things we're trying to do. We're trying to learn how to prevent device infection with some of the surface coatings that, the example I showed is one of many. We're trying to learn how to treat an infected device. This is no simple task. How do we determine that a device is actually infected? And once it is, how do we clear that infection? And then finally, how do we cure the device infection altogether? And this really brings in questions about personalized medicine and things associated with that. We've already done pretty substantial team building, and this is one of the exciting things. A group of us got together last October to kind of kick things off, and we had our so-called first discovery workshop last February, and we were just starting to build momentum, and then the whole thing kind of fell apart with the COVID crisis. But you can see a growing list of stakeholders who've been involved, and obviously there are a lot of industries who are excited about trying to resolve these problems. So with that, let me just conclude. If the details of my research are interesting, there are opportunities for collaboration, or if you have ideas or thoughts about this big, hairy, audacious goal, I've put my email address. I'd welcome any interaction or comments.
Well, thanks much, Matt. Very exciting work and very bold vision in terms of how to indeed prevent implant infections going forward. I don't see any questions submitted, and then let me try and ask one, Matt. So what are the regulatory hurdles that one would have to overcome in order to bring your discoveries to clinical applications?
Well, that's a $64,000, probably more like a $64 million question, Henry. The regulatory hurdles are substantial, and not because they. Well, it's because there really aren't established pathways yet for dealing with infection, and the mindset is still we need to approve these devices in the traditional fashion. So when it comes to proving efficacy and safety, that means a clinical trial. And if you think about something like a hip or a knee, where the infection rate is 1 or 2 or 3%, that means that in order to get a few hundred people who are infected, you need a sample size that is probably about 20,000 patients. And so it's been just prohibitive from all parties involved. And part of the challenge associated with this roadmap exercise is to find out ways to both engage the FDA and as well as work with the FDA to help them establish policies and standard practices that might facilitate the qualification of some of these devices. And just in light of everything we're seeing now with COVID and the vaccines and the treatments, it's clear that under the right circumstances, changes to policies and procedures can be made. And that's really an important societal aspect of this overall research field.
Okay, great. I have quite a few questions submitted online, and in the interest of time, let me just convey one question and then we'll move on. Given the constant presence of the antibiotic, can you comment on any propensity to foster resistance to the antibiotic?
So I think the question is getting at, the heart of the issue of why the FDA and others are very reluctant to include antibiotics into these devices. Because again, in the case of the bone cement, for example, this is used increasingly, and it wasn't meant to be used as frequently as it is now. So if the infection rate, say, is 2%, that means that 98% of the patients are having antimicrobial eluted and doing something in the body promoting resistance. So the idea behind these self-defensive surfaces is to only allow antimicrobial release when and where it's needed. I should say, just on a second note, there's a whole category of research out there that I'm not directly involved in, trying to find things like antimicrobial peptides or antimicrobial peptoids, which don't have the same resistance generating mechanisms that some of the traditional antibiotics have. So, there is perhaps some light at the tunnel, albeit far off.
Okay, thanks.